Hello, dear learner, dear viewer. Welcome back to our videos, Smart Learners Studio Kiongozi. This is yet another video whereby we are going to discuss something very small about biological drawings. So look at the handwriting, look at the wordings there, the principles of making biological drawings. So I have 9 points that you should take seriously when you're preparing to make a biological drawing. These points I would wish that you note them somewhere, and please make sure you keep on following our channel for more and more knowledge from our side. Point number 1, look at the red wording. Important points to note when making biological drawings. 1, use a well-sharpened pencil. Write with me. Point 1, use a well-sharpened pencil. Some learners will use ink. It is not allowed. Just use a well-sharpened pencil when you are making a drawing. 2, the diagram should occupy at least half. The drawing should occupy half or 3/4 of the space provided space. At least half. Make sure that the space you provided is well adequately used in drawing that drawing or making that drawing. Number 3, each drawing must have a title. Each drawing must have a title. Title for each drawing. For example, I will give you 2 examples of drawings that we will make. So we must have titles for the drawings. Number 4, enough space around the diagram. Leave enough space around the diagram or the drawing. This is, for example, this is because you need to label it, and as you label, you must have enough space so that the labels can easily be identified. So around the diagram, I will still show you when I give you an example towards the end of this video. Number 5, avoid double lines. Avoid double lines. Avoid double lines when making outlines. When making outlines of the drawing, just avoid double lines when making outlines of that drawing. Number 6, avoid arrows. Avoid arrows for label lines. The label lines, there should be no arrows when you are labeling the drawing. Number 7, never shade the drawing, the diagram. Never shade the diagram. Number 8, labels should never cross each other. During labeling, ensure that no label line crosses any other. Number 9, the last one, calculate the magnification of the drawing. This one, I know we have talked about drawing magnification, drawing magnification. You can apply the formula that we discussed in that video of magnification. So after giving you those 9 points, I would wish that I give you at least 2 examples of diagrams or drawings that and how you're supposed to make them when you are in a situation, maybe in an examination, and you need to make a drawing. The first one is going to be a hand lens. I will give you 2 examples. I will give you example number 1. The first example shall be a magnifying lens, a hand lens, So that is my title, the hand lens. I'll just make a simple diagram of a hand lens, and then the second one shall be an animal cell, under the viewing of a light microscope, So let us see how you make these drawings, and of course that is in reference to the principles of making biological drawings. My sharp pencil is here. Of course, I must also have a rubber and just a simple one, a hand lens, magnifying glass. So this is what you do. Look at the amount of space that I'm given. At least I should use more than half, and this is what I come up with. That is what I come up with. I'm drawing it. I'm using a free hand. That is the frame. So in case you have your rubber, you must ensure that you are a bit accurate. Like that. Yeah, something. So you look at this, we have this one needs to be clear. This is our hand lens. Using a sharp pencil, and ensure that your diagram is big enough in the space provided. So I'm almost done doing the frame, and all the way as we go to the handle. Yes. Some students call it hand, but it is wrong. It should be the handle. Drawings that we make in biology must be done using a free hand. Now let us label it. We have used more than half of the space that we are provided with. And now the next thing is labeling. We shall use horizontal straight lines, probably use a ruler. We start from this out part here. Use a straight label line like that. Then we label it. No arrows. Remember we said that in the principles, no arrows in the label lines. So this one is called the frame. The next one is the one that follows it. This bigger part here. We can label it on our right. Again, a straight label line like that, because we said no label line should cross any other. This one is the convex lens. convex lens, and maybe the last one is the handle. This one here, this one is the handle. So this is how you make a biological drawing. All the principles are followed. Example number 2 is the animal cell. When you are viewing it under a light microscope, remember in the viewing of an animal cell in under a light microscope, you will not see details of that cell. You see quite very few number of organelles in that. So it is irregular in shape. We can also make that drawing. This is an animal cell that only has the cell membrane. Cell membrane. Just want to make it clear for you. So, making good use of my rubber. This is the outer part of the cell, which is the cell membrane. The lines must be continuous so that at least the diagram, the lines become proportional across. We shall see 3 organelles only. 3 organelles or structures, intracellular structures within this cell. The first one is what we have done. The second one is the centermost one. Centermost one. And the third one is just at the middle. So we have used more than half of the space that we provided with. Now the next thing is labeling our diagram. Again, we use horizontal straight lines. We start from the outermost one. This is the cell membrane. Cell membrane. The second one is the middle one, which is the cytoplasm. Cytoplasm. And finally, the most middle, we can take this arrow downwards like this just to ensure that they don't cross each other. This is the nucleus. The nucleus. Just check on the spellings. So that is our second example and the last one. If you look at those diagrams, they are neat. We have used more than the space, more than the half of the space that we're given. We have used a sharp pencil both in drawing and labeling. And so learners, keep following me. I am your teacher, Mr. Wilfred. See you in our next video. Bye.